For this project, I will be covering cystic fibrosis with a later inclusion of cholera for reasons that will be covered later. Cystic fibrosis is an autosomal recessive genetic condition which causes changes in the secretions such as mucus and sweat. The changes in mucus in the endothelial linings is the most relevant for disease. The change in the mucus within the lung causes difficulty breathing, and changes in the mucus within the GI tract creates problems with digestion and nutrient absorption. These changes are caused by a mutation which reduces the functionality of the chlorine and bicarbonate channel protein, which causes changes in water movement along in the endothelial cells. So, cystic fibrosis itself doesn't actually have anything to do with cell signaling because the mutation that causes cystic fibrosis is within the channel protein itself, not within the signaling pathways that control the opening and closing of the channel. So, uh, inc- for the non-functioning mechanism of this pathway, cholera will be used instead. So, in the healthy signaling pathway, the cystic fibrosis transmembrane conductance regulator, the channel protein which allows chlorine and bicarbonate, is controlled by protein kinase A. Protein kinase A, both through the phosphorylation of consensus sites on the CFTR and direct interaction. The cystic fibrosis conductance regulator has several consensus sites, some of which activate or inhibit the channel, which can also depend on which other sites are phosphorylated or dephosphorylated, as well as pKa binding directly to CFTR, having giving pKa binding directly to CFTR, having the capability to open the channel while it is binded. Uh, CAMP, which activates pKa, is regulated through the activity of adenyl cyclases and phosphodiesterases. The adenyl cyclases which produce CAMP are regulated through G protein subunits, which are in turn regulated by GPCRs, mostly adrenal receptors. CAMP is controlled through negative feedback through the phosphodiesterases because higher levels of CAMP leads to the opening of calcium channels in the cell membrane, which increases cellular concentrations of calcium, which in turn increases the activity of phosphodiesterases, causing a drop in CAMP. The diagram on the left shows a CAMP pathway, though not specifically for CFTR regulation. And the diagram on the right shows the CAMP pathway for the regulation of the opening and closing, uh, short term of CFTR, as well as a long term uh, regulation pathway where a signal from outside the cell can cause the creation of interfering RNA, and through Argonaut, it will block the translation of the mRNA that encodes for CFTR, which is an example of post-transcriptional regulation. So while cystic fibrosis does not actually have anything to do with disease defects within the signaling pathways, cholera toxin does. Cholera toxin is a two subunit toxin. It is not a homodimer. The two subunits are different. There's an A subunit and a B subunit. The B subunit binds to a ganglioside in the cell membrane to transport the A and B complex into the cell, where the A subunit interacts with the alpha subunit of the G protein, which is normally the subunit of the G protein that binds to adenyl cyclases and activates them. And under normal conditions, the alpha subunit binds with the beta and gamma subunits, either to turn back into into an inactive G protein, but the A subunit of the cholera toxin binds to the alpha subunit of the G protein, preventing it from turning back into an inactive G protein, which keeps adenyl cyclases activated, leading to elevated levels of CAMP, pKa, and then the continuous opening of the CFTR channels. The open channels cause a loss of ions and subsequent loss in fluids, causing diarrhea and the dehydration that leads to death and cholera. Uh, yeah, those are just references.